Hello, good afternoon, everybody. Let's start the second round of our presentations and discussions. The first speaker is Siguna Müller. She's a mathematician, or at least her first PhD is in mathematics, in particular oriented to cryptography. But then she moved to biology and she got a second PhD, both in the second one, sorry, in the United States, in the United States, and then she worked for a while in a biology laboratory, and finally moved back to Austria, her own country of origin, where she is an independent researcher, and I think she is independent, very deeply independent. Siguna.
Thank you, Giuseppe, for these kind remarks. And it's such a pleasure and honor to be here and give this talk to a wonderful audience. I feel so much has already been said. And well, let's jump in. Giuseppe, please, at some point, give me a heads up. As you all know, I probably have too much material. So about 10, when I have 10 minutes left, can you please let me know? So as Giuseppe said, I started out in mathematics, where life can be really easy when it comes into causality, because there it's very simple. When you talk about cause and effect, you typically have a statement of if certain criteria are satisfied, then so-and-so is supposed to happen. And just to get us started here with this famous example of the rabbits multiplying, giving rise to the Fibonacci numbers. That's just one typical example where you start out with if, you have a pair of rabbits, and then this is how often they multiply and mate, and when they give rise to babies, and so on and so forth. And then you observe after n generations, or here in this setting, like they give rise to new babies every 1 month, which they call an idealized setting, question mark. Then you have that many babies, and then you can count how many rabbits you have. And that gives the famous Fibonacci numbers, which in mathematics, of course, then we go on and we need to prove that this is really a relationship that always holds true. Now, what is implicit here? And, as I said, math likes to work with an idealized setting, which of course is not what we observe in biology and nature. Because, if you look at what they call idealized is, those rabbits just keep multiplying and multiplying and multiplying, never die, and so on and so forth. And implicitly, what is also not asked is, the key players involved. So who, what, how, what's going on, where, all of those things are, pretty much ignored. However, during this talk, it's those questions that will be guiding many of the considerations. Now, so the previous Fibonacci sequence is intimately related to, or pretty much equivalent to, what is also known as the golden ratio. And it's a famous quantity that is, obvious throughout of nature. And, you can see it in the way that galaxies are forming. If you look at the spiral, the same things are how hurricanes are forming. You see it in lots of flowers, trees growing. I guess I could fill many slides with this. And indeed, many people have devoted their entire career to studying those numbers. And I too spend a lot of time studying generalizations of those relationships. What is important about this, that once you have this relationship, this what is called the golden ratio or generalizations, they typically not only imply something that aesthetically looks appealing, But if you look into it mathematically, you can actually show that there are some minimality conditions going on. And this seems to be at the core of why you see the same thing. You know, I'm not saying exclusively, but you have this phenomenon here, and you see this in many different settings. I call this a hallmark of causality. It's this very unique, distinct characteristics that you see wherever you look. And again, that will be one of the main things to keep in mind for this talk, this unique pattern wherever you look, the same pattern. So to switch gears, what happened in 2020, and I have to be very honest here, early 2020, when the pandemic first broke out, I was an unbeliever. You know, I was very much what now would call a conspiracy theorist, although I was told the only difference between conspiracy theory and conspiracy is 6 months. So anyway, I didn't think, for instance, that the virus really existed. It was just too much for me in the beginning. So I went through all sorts of denial attempts. And then this happened. I saw this paper. And the paper said, well, they had some group of researchers had reconstructed the virus, synthetically, starting from their genetic information alone. And that was way early in the pandemic, rather early, during a time when there was no viral isolates of the novel virus available yet. And here you can see some of their pictures, how they did this, and you know what this virus is doing in different cells. So, oops. So that kind of was a very strong, convincing argument to me, and also how they did this. That they literally started off with the genetic sequence information. Now, this is actually a technique, what I learned later, that has been around for quite a while. It's called infectious cloning techniques, or infectious disease cloning. And you probably know it from the most popular article was by the Craig Venter lab of the, what I described, the generation of an artificial bacterial cell. Now, what is striking me here, and this is why I'm bringing it in this context, is as the title of this article says very explicitly, creation of a bacterial cell controlled by the genome. So if this is not the central dogma rearing its head, I don't know what is. And so really what they do, they start out with your digital DNA and then chemically synthesize your DNA, and that you can assemble in fragments in yeast or in another carrier. Now, if you are working or constructing RNA viruses, you would just need another step, another polymerase, some enzymes that facilitates the conversion from DNA to RNA. And then you can go on and, you know, get the more or less viable virus out of this. So that's the first thing. And the second thing is that the same approach is actually being taken with vaccines. And again, the main starting point here is you start out with not the entire virus, you start out with the synthetic sequence, with the sequence that is from the epitopes, from the immunogenic protein. And from then on, you start to create your vaccine, and you pretty much ignore the rest. So again, a very causal approach. You know, the driver here is the DNA or the genetic sequence. So you don't really work with cell lines or anything, which, you know, is something that's been celebrated, as they say. Now here to the right, an example of I think that's Pfizer, the beginning of what is going into the vaccine. This is literally the starting of the vaccine with some modifications here, some chemical modifications, but it's really the code that goes into a DNA printer, and out comes, some molecules that are further modified chemically. You get your RNA, and then you stick it into your lipid carriers, nanoparticles, and off you go. But those are the key things here, that at both of those key components of the pandemic, the virus itself and the vaccine, you have the starting point, the genetic information. So based on this, my aim is to really analyze various causality issues from this angle, based on the sequence information. So the one challenge here is, if you look at sequences as believed causal determinants, well, are we really looking at the right players? So it's always good to start at the beginning. So looking at the origin of the virus itself, here's this paper that said, well, what we know that the virus docks to the ACE2 receptor in humans. And so the ACE2 receptor is also in other animals. So let's just look as to which other animals afford which binding, and develop through their binding affinities a hierarchy, and perhaps that can tell us who would be the missing intermediate that has given rise to this virus. So what they did, it was based on in silico modeling, that they just compared the spike proteins behavior from different, species that have been in question to, what degree do they bind more or less to the ACE2 receptor. Well, the first surprising finding was that by far the highest binding was to the human ACE2 receptor. Now, in context, what's even more interesting, perhaps, is that they noticed.
Siguna, may I interrupt briefly? We are losing you on audio.
Yeah.
Can you turn off your camera?
Can you turn it off for me, please? I don't see the icon at the moment. Shall I continue? Is that better? Hello?
Continue, please.
Okay. So, so the surprising finding here was that sequence information does not tell you a whole lot. In fact, it's very much in contrast, as if you look at it based on structural determinants. So it could go then either way and go all over the place. In other words, the sequence information gives you the opposite of what you would find from if you were to look at sequence information. And so which raises a lot of questions. I mean, for once, they were only looking at the ACE2 receptor. But I guess the other question is, and I should say there's other models in studies that show that entry into the host cell is supported by other methods too. But then the real question is, how do we evaluate all this? How do you know? I mean, obviously sequence information alone is not enough. I have all sorts of other interactions, hydrophobic, electrostatic, hydrogen bonding, and on. So, I mean, that raises just more questions, showing that the sequence-based approach really cannot be used in this context. Now, let's go back also to the pandemic. Another issue was that there were no safe vaccines for coronaviruses. And the main problem was that of antibody-dependent enhancement. And the situation is that what was observed, and I should say it was observed, in various studies, in vitro, monkeys, other animal models. I forgot to write here, actually, also with humans, with various viruses that with these types of vaccines, people developed initially a wonderful antibody response, but once they were challenged with the true virus, children died and all sorts of nasty things happened. So dependent on the antibodies, antibody-dependent enhancement. And one of the studies that I'd like to focus on here for a second is this study was also before the pandemic. And what they found just blew my mind. And so what they did here, they looked at SARS, so the first coronavirus, or SARS-CoV-2, and they looked at various peptide sequences that are recognized by SARS-infected patients, so the epitopes. And they then cloned the various antibodies and tried to see a certain relationship. Now, for once, they found that there was really no one-to-one relationship. Again, so if you were to start out with the sequence information, you quickly get into trouble. Now, the problem here is not only that, different sequences are being recognized by different antibodies, but the real problem is that you get the different type of functions of those antibodies. Some actually give you protection to the disease, the neutralizing antibodies, and the others are the ADE type antibodies, which enhance infection and actually make disease worse. Now, why am I showing you here this part? I found this absolutely hilarious because you probably won't be able to see this. I'm sorry, it's a copy. The green part here is, so on the one hand, you have an epitope that gives rise to a very nice neutralizing antibody. I don't know if you can see my mouse here. And if you look at the other one, it has just a few amino acids added to it. And it changes the behavior from triggering neutralizing antibody response to an ADE response. So that really caused me to ask, so what on earth is the immune system really reacting to? If you have almost the same here, and then you have this little change, and then you have the completely opposite response, what on earth is happening? Now, in what we hear these days is everyone is focusing on these IgGs, or the IgAs, and all the immunoglobulins, which sit at the very end of a very complicated immune response that I'm not going to bore you with, because it's extremely complicated and we would be here forever. But would please keep in mind that normally you would first have the innate immune response if we actually have an adaptive immune response. So back to those main players that everyone is looking at these days. In the adaptive immune response, the question is, do we have the important players? And as is known for a while, but perhaps even challenged today, is adaptive immunity, is believed to rely very much on the amino acid composition. And so to give the different antibodies a few months ago is, oops, there's much more going on than just gene sequence determined. And so, for example, I depicted this as a hierarchy, which really isn't meant seen as a hierarchy, just initially to say there's different layers. What people typically look at is, amino acid sequence-based determinants. And what I figure is that what the virus is doing is being mirrored by our immune system and vice versa. And if you look at coronaviruses in particular, they just don't only have, you know, their features are not just being a coronavirus with a spike protein, it's a glycosylated protein. So you have modifications that very much impact how the virus responds and escapes our immune system. So layered on top of the amino acid base, you have other strategies like post-translational modifications. You add some sugar molecules. Oops, here is the viral site. And conformational changes, masquerading, cofactors in addition to glycans, and so on and so forth. And ironically, it turned out that the immune system is really able to counteract and mirror all of this. Now, what this means is you have these non-genetic antibody modification strategies that can even overrule the more traditional ones based on the amino acid composition alone, but they are much more refined because they are involved all sorts of post-modification modifications, are subject to up and down regulation, whatnot. I mean, it's very, very variable and flexible, you know, as opposed to a singular pattern, which I said to keep in mind as a causal driver of some causation. Now, of course, the other thing is with those antibody diversification mechanisms is that they, unfortunately, well, the strange thing is sometimes they're only used as a last resort mechanism. Sometimes they can escalate. And that's known from other virus studies with viruses and lead to severe or disease or even death. Now, perhaps corroborating this was this finding of a recent article. They looked at, well, post-mortem analysis of COVID deaths, and they found something really striking. They looked at the presence of the virus in different tissues and organs, and you know what eventually was it that killed those people. And to their surprise, there was a huge disconnect. In other words, you have both. You have sometimes viral particles present, but there's not a lot of inflammation going on. On the other hand, you have inflammation that's eventually believed to be what's killed those people, but there is not a lot of virus there. So this disconnect caused those authors to believe that fatal COVID in particular is not really caused by the virus, but more by the immune response. And, you know, if you go back, or, you know, what I've been trying to say is our at least adaptive immune response is so flexible, interacting all the different layers and players and whatnot, and very much depending on the pathophysiological state of the host. So it would not be surprising that this flexibility is what either could lead to health or unfortunately the other effect. But here, the hallmark, we have a lot of flexibility. Now, on the other hand, what we are observing on the global level is the complete difference of flexibility. Is the entire world seems to be under the grip of Delta. More and more, it has become the dominant strain of the virus pretty much everywhere. And if you look at the global patterns, there's lots of patterns. I just grabbed the first few. In fact, here, Richard Fleming added the time points of when vaccination campaigns started. And you can see that here is the different strains of the virus in different countries. Initially had a whole lot of variation of different strains. They've always been present, and then suddenly you have fewer variation. And I mean, fact really is, when vaccination started, they were really geared towards the original virus, and not really against the others, because they developed later. So what you have here now is, here the green is the Delta, and you see it pretty much, well, eliminated the others. So what certainly is true, that this Delta is known to impair the efficacy of vaccines. But, you know, I would suggest, and I know I'm not alone in this, that it's actually also the other way around. It's because you had those leaky vaccines that they were driving the development of variants of concern. Now, what do I mean by leaky vaccines? There is a paper that, or a study of a few years ago on chicken. And they showed that if you have a vaccine that doesn't completely kill the virus, but still allow transmission, you allow the pathogen to survive much longer in its host. And this way it give rise to more concerning variants. And notably, that's the current COVID vaccines are by design leaky vaccines. So I'm not going to go, there is a lot of different studies that really try to find a causal relation from the rollout of the vaccine and an immune pressure that's being caused globally to drive the Delta variant. So I would like to go on to something more curious, at least for me. The immune pressure, also how we see it in non-sequence-based determinants. And I found this very interesting. This paper, I forgot the site where it's coming from, was published in October, so very recently. And well, we all know that this unique insert here, the PRRA insert at the furin cleavage site, is unique to the new virus, and that's really what's making it so much, more of a concern. Now, the surprising thing is that this cleavage site, or I should say, furin cleavage, is actually modulated by host cell glycosylation. So one of the post-translational modifications that I was also looking at in the last few months or last year. And well, this is what's happening with the wild type virus. And what they found is now there's mutations out, or at least I should say that the more concerning variants, they have mutations here that go in hand with a change in the glycosylation pattern. Now, originally with the wild type virus, the glycosylation actually acts as a buffer, so making it harder for the virus. Now, with those changes, the virus is actually getting an advantage. And my feeling is that with the wild type virus, the host is allowed to mount this very variable and flexible response, but vaccines are actually designed in a way that exactly at this position, at the corresponding glycosides, the vaccine actually makes a little bit of a change and fixes something. So naturally you have variability. Here you have stability because of the vaccine. And I feel this is actually what might be driving is too strong a word, allowing some escape for the virus. Now, here's some other curiosity, at least I found mind-blowing for several reasons. Again, also very recent, in October, publication. People were looking at people in hospital system, healthcare workers, and what the conclusion they came to is some people never, they clear COVID infection so quickly that they don't even test positive. nor produce antibodies. And the idea here is that this protection is afforded by memory T cells that were produced from the common cold coronaviruses, which brings me to the question that I want to pose is, can it be possible that it was actually the other players that we have ignored, the T cells, the innate immunity, that are more important? So perhaps to combat COVID, they might be plan A, and perhaps antibodies are only plan B. And then antibody diversification, as I said, is often a last resort mechanism. It's also very expensive to the organism, could be plan C. Now, so let's look at the antibodies to prevent disease in the context of causation. And it's paradoxical, because the current model is that, you know, it's believed that the more antibodies you have, the more you're protected. And there's a number of interesting counterexamples. For instance, you see higher antibodies with severe cases, and patients who recover quickly have no or low levels of antibodies, and all sorts of other strange phenomena. So one of the key concerns I have is antibody-dependent enhancement, also in the context of non-sequence-based determinants. ADE, as I said, has been a concern for a while, and even prior to the pandemic. And in 2020, it appeared as there was no evidence that ADE, antibody-dependent enhancement, would facilitate the spread of well, the first coronavirus. And the model that they were developing then is, the model is, well, if you have the virus, normally it gets identified by your antibodies, and then your immune cells take care of it, you're done. Now with ADE, what's going on, they identified a very specific mechanism, is that those antibodies actually hooked on at the other side into a different cell, and then allow you to enter other cells and infect other cells. And they had very specific mechanisms, and the idea was, well, that's the FC receptor-based mechanism is pretty much it. And since it wasn't observed in SARS-CoV-1, everyone thought we were good, but I think we're in trouble here. So what suddenly is more and more emerging is that there is, well, this is not a causal one-way relationship. In fact, we do not know what really causes antibodies to become ADEs, or to trigger enhancement or to be protective. In fact, in my one paper, I asked a lot of questions that we don't know about antibodies, when they could turn into ADE or facilitate this enhancement beyond the sequence-based determinants, but more on, oh my gosh, I cannot go into this. It will take too long. Fact is that a lot of this is actually, unfortunately, being discovered now. Some fairly recent paper, June 21, they identified a new mechanism for antibody-dependent enhancement, ironically, for those more severe patients. And here is the sad thing. It seems to be some selection going on, that severe patients seem to have more of these bad antibodies. And on the other hand, at the same time, the scale seems to be tipping, that the more the coronavirus evolves, the more it seems to give rise to those unpleasant, dangerous antibodies. So the newer versions of variants of SARS-CoV-2 have a higher affinity for those bad antibodies. Here are just a few examples. I guess I'm not going into detail here. New publications all showing new players. So antibody-dependent enhancement, not only relying on the sequence-dependent mechanisms that has previously been known, but involving new players, new mechanisms.
Una, you have 10 more minutes.
Great, thank you. And I think this is the important thing I wanted to get to, because when you talk about causation, one of the main things we really want to get at is what type of action is being triggered. And in this case, I guess it was the action of the protective immune response. And the idea is being that the causation in the sense of an antigen triggers the protective immune response. Well, here's a few counter examples. I would think they are kind of sobering. Despite being designed to actually work on the adaptive immune system, in particular because COVID vaccines really trigger antibody production. Some recent studies show that the COVID vaccines can actually alter our innate immune response. What is meant by this, that they could lead to what is called tolerance. Tolerance is when you then encounter other viruses or pathogens, that the immune system no longer responds. And if I, would like to remind you that typically we would expect that the innate immune response would have to fire first to get to an adaptive one. So the effects of this are not known. It's also not known how those vaccines interfere with other vaccines. And perhaps the most, even more, I don't know if I could say even more, just equally concerning is a very, actually it's not so recent, 8th of October publication. I saw it about a week ago and it really floored me. What they claim is that the spike of the coronavirus itself damages various DNA repair mechanisms and inhibits the VDJ recombination. What they did, they showed this in vitro studies. What they were really looking at is at the adaptive immune response. And they observed, well, let me just say what they observed. So what is believed is that spike proteins normally are only in the cytosol. What they found, however, is that various proteins of coronavirus are also detected in the nucleus, including the spike. And what they then did in their in vitro studies is that they found explicitly, I think this is rather sobering, that the very presence of the spike protein in the nucleus inhibits major DNA repair mechanisms. And one of those or two of those are implied with these mechanisms that's responsible for determining the diversity of the antibodies, the regions. However, what is concerning here is it's an in vitro study. And the authors themselves, they asked like one quick question, hey, could this be important for vaccines? But they didn't go any further. I would like to go further and ask this as one of the final question here. Although their study was only in vitro, what do we know from the virus? The virus normally doesn't go all over the body. Although what we heard earlier, of course, there is the possibility of exosomes and also other vesicles that can carry those material to various places, including where you have those mechanisms taking place, the VDJ recombination, which is in the lymphocyte precursors. Now, with vaccines, normally they should stay in the muscle. And what really is the concern for me is because, as we know from several studies, the vaccine does not only stay in the muscle. So not only is it that the effect, the action is not so causal and singular as believed, vaccines have all sorts of additional contributors and helpers as elements, like nanoparticle carriers that carry those vaccines pretty much everywhere. You might not be able to read this here, but that is including the thymus, the spleen, the bone marrow, everywhere. So this leads to the concerning realization that yes, the vaccines could actually reprogram also not only the adaptive immunity, but also more importantly, I think, or equally importantly, the double-strand DNA repair mechanisms. So that was a question of causality in terms of where those things go. I would have had another quick section, but I'm timing aspects, what and when is happening in terms of, we talked about this earlier a little bit, some other aspects I don't see yet any answers in the literature, but interesting questions that I would like to ask. One is the concept of original antigenic sin. Is if the immune system learned something in the past, it gets stuck, how does it get unstuck? What I find even more concerning and is I've done some work on this just theoretically in the past on organisms that have no central nervous system, like slime mold, you know, and they can actually learn. I know simple amoebas, they can learn by simple repetition, and it's a form of associative learning, which we are all familiar with, is Pavlov's dog, you know, ringing the bell and they start salivating. And it seems to be something similar going on in terms of some network dynamics with gene networks still firing and going on, even if you were to remove the stimulant and so on and so forth. A lot of questions. I have no idea what this leads to, autoimmune responses, how hyperreactive responses, and so on and so forth. So no answer here, just questions. So altogether, I think I've given you a number of, well, not causal aspects, but non-causal aspects of where if you rely on sequence-based variants, you don't see causation, you see numbers of counterexamples. So my first conclusion is we really need a vaccine that goes beyond the sequence-only approach and looks at the virus as a whole. And I guess my second conclusion, my claim, it's not the virus that's causing the pandemic. What's really at the core of it is our naive understanding and incomplete understanding of nature, are trying to base it all on a very small conception and then working with the lock and key approach of the antigen antibody determined by the sequences. That's just not it. So sorry for taking so long. Thank you.
Thank you, Siguna.